So, good morning, everyone. Today, we all will talk about the laboratory safety units and measurement. Laboratory safety. That means here we are going to talk about the practices which we are going to use into the laboratory as well as the things required in the laboratory. That means we require to do the lots of calculation and the measurement. Now see, we are going to discuss the entire unit. So all the lectures will be kept under the same playlist. So you just go through with the playlist. So whatever topics are related to this particular unit will be added in the same. So the content of this unit is Introduction to Good Laboratory Practices, that is GLP, then use of safety symbols, then unit of measurements. Unit of measurements and biostatistics are the two different things. In unit of measurement, we will be talking about just about units like unit used for measuring the temperature. Then thereafter biostatistic is the another calculation part will be discussed into the same unit. Now good laboratory practices, that is GLP. So what is GLP or what is good laboratory practices? So these are the set of principles that provide a framework within which laboratory studies are planned, performed, monitored, reported, and achieved. So see, it is very important to remember the definition of it, that GLP, that is good laboratory practices, is a set of principles that provides framework blueprint in within which the laboratory studies are planned. Whatever we are going to study into the laboratory, then whatever we are going to perform in the laboratory, as well as the monitor, what we are going to monitor in the laboratory, reported and achieved, and maybe maintain the proper records of the laboratory. Now see, when after the lockdown, when you will join the college, so you will find that into the laboratory we maintain the students muster. You have the biometry also. So along with the biometry, the another record is maintained. So why? To understand or to find out whenever there is an inspection, to be check the footsteps, that how many students have worked in this laboratory in the entire year. Actually, the GLP is FDA regulation. The full form of FDA is Food and Drugs Administration. Remember the food, this full form of FDA, that is Food and Drug Administration. It is the Department of United States of America. In the early 70s, it is observed that the number of fraudulent activities and incorrect laboratory practices occurs into the many companies into the US. Means how it started, a small story is there behind it. What happened that the to reduce the cost of the cosmetics, you can say the pharmaceuticals, so they started doing the fraudulence, they started doing the incorrect practices. They used to carried out the wrong, means they used to give the wrong information. There is one example which brings the GLP in action. What happened? Once upon a time in the US, one company, they checked, uh, actually there were 40 company indulged in that activity, but one company, it was found that company was the pharmaceuticals and cosmetic company. So they were doing the research, means you all might be knowing that whenever we are doing any type of means practical Anything before coming to the market is tested. Now, if someone has to introduce any type of medicine into the market, it is very, very compulsory for them to check it on the animal. So likewise, they were checking on the animal. So previously, they used to do the work into the company only. So what happened? They were checking it on the rats, and they found that the rats developed means they developed the cancer, and because of that, many rats died. But the production company, they did not disclose that their rat died and all. Then with the help means because of that fraudulence, the company has introduced the product into the market. And what happened, that product came into the market, and now definitely if rats are dying, rats are what, they are mammal only. So it is found that slowly the people were also dying. So they died, and because of this fraud, it led to the implementation of GLP in the US, and adherence it was made compulsory for all the industry and laboratories who are in manufacturing industrial, whether it is chemical, pharmaceutical, veterinary drugs, pesticide, cosmetic product, food product, feed additives, etc. So it is become very, very compulsory to follow. I repeat, which all are going to follow the GLP? It is going to be followed by all industry and all laboratories which are in the manufacturing industrial chemicals, pharmaceuticals, veterinary drugs, pesticides, cosmetic products, food products, and the feed additives, etc. So to facilitate the ensure the GLP by majority of the world, they form one organization that is called as OECD, Organization for Economic Cooperation and Development, took place of all interested countries who wish to do business with the US organization. Means if you have to do the business into the US, so you have to follow the OECD, that is the Organization for Economic Cooperation and Development. So this will ensure that the company is following the GLP, and we know that it is under the regulation of FDA, that is the Food and Drug Administration. Most of the time people get confused. GLP is what? It is nothing but the standard of laboratory safety, like wearing safety goggles and all. Absolutely not. The GLP is something which is following and recording the chemicals which are in contact with the human or any animal. So we should not confuse. Now, what is the India status? Now we understood there was fraudulence into the US. 40 companies were found guilty, and they were giving the wrong reports, and because of that it came on effect of the health. Then GLP has been started. Then GLP was standardized as OECD, and thereafter the US has decided if any company wants to do business with the US, they have to follow OECD. Then thereafter it becomes standard and international. Now you will find that whenever we are buying some products or trademark FDA, if it is related to the food, so food or drugs, FDA and some kind of certification is on that. So it is nothing but the monitoring body to which these companies are submitting the reports. Now let us discuss about the India status. So India is a full-time member of the Mutual Acceptance of Data OECD. Hence, the data generated by Indian GLP certified laboratories are acceptable in all the OECD member countries. Understood? So means we have taken the OECD full-time membership. So whatever things, whatever work is done in our laboratory is worldwide accepted to get GLP certificates in the India, the test facilities, laboratories have to submit application form along with the prescribed fee to the NGCMA. Means suppose you are starting a business, okay, and that business is the industrial production of anything, pharmaceutical, food, veterinary, cosmetic. So you want to definitely, you will allowed to sell your product into the market only and only you will get the GLP certification. So now if you want to apply in India for the GLP certification, so you have to submit one application form. Now along with the prescribed fees, whatever fees is there for the GLP, now you are going to submit this fees to NGCMA. I repeat, it is NGCMA, National Good Laboratory Practice Compliance Monitoring Authority. Understood? So once you applied on it, they will check your company, then they will check your product, and they will check your laboratory facilities, and thereafter a pre-inspection of the laboratory will be done. Then after inspection, the inspection team and the report is submitted, which is considered by the technical committee. They will check on the basis of their recommendations. Chairman NGCMA will take the final decision whether your company should be given the GLP or not. Then GLP certification is valid for the period of 3 years only. And GLP, see this point to be remembered, the GLP certificate is not lifetime that XYZ company is having the GLP. No, you need to renew. That means after every 3 years, the complete procedure, application, inspection, everything will go on. So GLP certification is valid for the period of 3 years only. The GLP secretariats organize surveillance at the end of every 18 months from the date of certification and a reassessment during the third year for maintaining the certification. That means if you want to go for the certification again, you want to continue with your this company, so you have to apply again, then again reassessment will be there. Although GLP certification is voluntary in nature, it is essential criteria for the export of any of its product by Indian organization or import any raw material from abroad. Means we feel like that GLP certification is only for bringing our product into the market. No, it is absolutely not like that. It is a very compulsory, it is voluntary certification, and it is one of the most important or essential criteria for the export. If you are not having GLP certification, you will not allowed to export your product. Not only this, if you want to import the raw product from the abroad, that time also GLP certification is required. Now GLP certification, where it required, so it applies to the non-clinical studies which is conducted for the assessment of the safety or efficacy of the chemicals. Now we had already discussed, it includes the pharmaceuticals, cosmetics. Then it helps assures the laboratory authorities that the data submitted are true, because they're checking your this data. They are checking your complete assessment. They will check your data, they will check your technical team. A complete inspection will be done. In our laboratory also, you will find that after few years they are conducting the audit. Audit is nothing but the inspection kind thing. So in college laboratory also audits are there, inspection is there. LIC committee is coming, they are observing that the laboratory is having the proper chemicals and all or not. So GLP, what are the objectives? GLP makes sure that the data submitted is a true reflection of the result that are obtained during the studies. Means sometimes anytime they can come and they can check whatever results you are submitting or not. Then GLP also makes sure that the data is the traceable. Whatever you are submitting is there or not there. Sometimes these bodies like FDA and all, They ask you to submit your product and they check your product in the laboratory also. So GLP promotes the international acceptance of the test. Now, what is the mission? So the mission is to test the system for achieving of the records. Then they check the apparatus, materials, and reagent facilities. They do the quality assurance programs, performance of the study. Then not only this, whatever studies are there, so they make sure reporting of the study results. Then standard operating systems. Now, for whom the GLP certificate is awarded, it is awarded for the following areas. Example, physical chemical testing. Physical chemical testing means generally whatever product is there, it's physical or chemical test. Then toxicity studies, mutagenicity studies. Note down the term mutagenicity. Mutagenicity means any substance which is bringing a change in the genetic makeup is called as the mutagen. I repeat, mutagen means a substance which causes the mutation. So what is the mutation? Mutation is any change in number of the chromosome or in the genome. So the substance which is causing the mutation is called as the mutagenicity. So they check whether the product which you are going to sell is causing the mutagenicity or not. Not only this, whether it is harmful for the environment or not. So environmental toxicity studies is also done. This will done in two areas, aquatic and terrestrial organism. Then studies on the behavior in the water, soil, air. Not only this, they check for the bioaccumulation, means whatever substance you are using, so it is not causing. You might have observed that DDT was a once upon a time very good pesticides, but because of bioaccumulation, means it remains into the organism for a longer period of time, it was banned. So they check for the bioaccumulation, that the chemical is not causing the bioaccumulation, biomagnification type of effect. Biomagnification means it is going to the higher hierarchy. Then residue studies, whether it is producing the residual effect. Residues means the remain of. Then studies on the effect of the mesocombs and the natural ecosystem, whether it is not harming the natural ecosystem. Not only this, analytical and clinical chemical testing is also done. So this is about the good laboratory certification, good laboratory practices. So this is for the industrial level. Now for you, what you are going to follow, you are also going to come into the laboratory. So see, there are 19 rules before entering into the lab. We make sure that you are going to follow these rules. What I am going to tell now is for you. The first thing is that first of all you need to remember that safety is everyone's responsibility. If any accident happens in the laboratory, not only your teacher, you are also responsible. Each and everyone is responsible. Your laboratory assistant, teacher, everyone is responsible for the safety. So whenever you are entering in the laboratory, make sure that you will come with the permission. You will not come uh, for like that only. Whenever you have practical, that time only you will enter. And before beginning of the experiment, you have to take the permission. Then before startup, you have to read the procedure carefully. Most of the time students come late, and you will, when you will come practically into the college, you will find that I don't allow any latecomer. Student feel offended that all the teachers are allowing, then why don't I allow? The reason is, if you have not heard the procedure, you can hurt yourself, and it is for your safety. If you don't know procedure, you should not perform the practical, because if you don't know, you can harm yourself, and it is for your safety. So make sure that you will always reach in the laboratory on time. Then if you are coming, if you are starting the practical, make sure that now in our laboratory we practice. First we explain you the procedure, then only we allow you. But then also if you have any doubt, before starting only you must ask. If an accident occurs, immediately you should tell your teacher. It is not like that, oh no, teacher will say this and that, whatever teacher will say, but you should inform. Then always you don't require to wear the goggles. Now see, some rules this are for you. The first thing is that you are compulsory have to wear your cotton apron. Now, it is very compulsory to wear cotton apron. Proper footwears you have to wear. It is not like that you are running into the laboratory with the high heels. I don't have any objection on the high heels or anything, but if you will slip, you will harm my 10 students. So make sure that proper footwear you will wear, and thereafter your head, either it should be covered, or if you have long hairs you need to tie it. Now you will tie your hair on the back side. Don't make your hair, if you have long hair, it is very compulsory for you to tie your hair. Now why I am emphasizing, first of all, I don't allow any girl, those who are coming with the open head into the laboratory, because if you will not tie, it can catch the fire. Because you don't know, we have the tables, we have 5 tables in our laboratory. So if your backside student is using fire, so that fire can catch to your long hair. So to avoid all such thing, avoid such accident, it is very compulsory for you to tie your hair very properly, or you can cover your hair. Most of the girls, they prefer to cover their hair because to avoid any type of accident. Now see, flowing sleeves. Nowadays it is in a very good fashion. Girls wear the girls. I have seen boys also. They wear the flowing sleeves. You should not wear any flowing sleeves into the laboratory. If you are wearing it, then you should tie it properly, because it can catch the fire very easily. Then gloves should be worn while experimenting with the toxic, then carcinogen, or any type of corrosive chemical. It is very compulsory. Gloves are kept into the laboratory, but if you are not getting it, make sure that you are carrying your gloves. You will not allowed to do any practical in the laboratory without the gloves, which contains the carcinogenic chemical. Carcinogenic means the chemicals which causes cancer. Corrosive, which causes the burns. Then volatile and inflammable chemicals should be kept covered and far from the source of fire when they are not in use. So whenever you are using any type of chemical, make a simple habit that you will cover it. Most of the time students take out the beaker and keep here and there. It should not be done. Care should be taken to prevent from catching fire, because you don't know the nature of the chemical. Then acids and alkaline fluid should be pipetted out using the rubber bulbs. We have the automatic pipettes, means which operated with the micro pipettes we have. Then we have the pipette pumps. So we don't allow, most of the time no chemical is allowed to pipette out. We have the pipette pumps in our laboratory, but if other students, those who are listening this, they should also follow it, that whenever you are may, pipetting out any chemical which is either alkaline or acidic in nature, make sure that you are using the rubber bulb. In our laboratory, in our college laboratory, we have the pipette pumps, so we don't allow any chemical to pipette out. Then eating food is not allowed. That you will not dare also into the laboratory. You should not talk and do the gossiping in the laboratory. No horseplay. You should not run. Then washing hands properly before leaving the laboratory. It is a very compulsory habit you need to develop, that whenever you are going out of the laboratory, make sure that before leaving the lab you are washing your hand. If you don't require burner, make sure that you will switch off it immediately. Then burner flame must be adjusted properly. Whether you want to use the oxidized flame or the reduced flame, make sure that type of flame is adjusted as per your practical. Then electrical appliances must be handled very carefully to avoid any type of shock. Then incubator, balance, centrifuge machine and other electrical equipments will not be moved from here to there. Wherever it is kept, you go there and you perform. Ma'am, all are standing here, my friends are there. No, wherever instrument is there, you will do. Then whenever you are observing from the compound microscope, make sure that you are using both the eye, because most of the time it is observed that students close their one eye and they focus from one eye only. So this is a very wrong practice. It can also affect your eyesight also. So make sure that you will open both the eye and you will observe from both eye only. Objective lens of the microscope must be taken care. Make sure that you will not lower down your objective cap while adjusting your microscope so down that it is touching the cover slip or your slide while focusing them. Now, most of the time, we are showing you the preserved specimen which are preserved into the glass jar. You should not touch the specimen. You should walk around the specimen. In our college, it is kept into the proper cupboard. So whenever we are removing it, make sure that you will not touch it, because the formalin used for preserving the biological specimen is carcinogenic. Just now I told you the meaning of the carcinogenic. It is cancer causing. Hence, contact of body parts should be avoided. You should not touch. Whenever you are entering in the laboratory, make sure that you have your rough journal. It is very compulsory. It should be maintained for recording the observation, whatever practicals you are doing, and it must be your regular practice. And then only you transfer it to your fair journal. Immediately, most of the students, they start writing into the journal. Who will copy it later? No, this is a wrong practice. You should maintain the record. You should avoid writing into the fair journal while performing the practical. Any chemical can fall on your fair journal. You have marks on your fair journal. Not only this, journal is the standard record of your whatever practicals you are doing. So we should avoid doing it. Then sometimes now in FY we don't ask student to dilute the acid, but remember this point: whenever you are diluting any acid, make sure first you will take water into the beaker and thereafter you will pour acid. Not you should go for the vice versa and dilute this, means pour small quantity of the acid and mix it with the glass rod. Accurate quantity of reagents must be taken. You should not, means if I told you that into the protocol 2 ml is given, no, I will take 3 ml. You will get the better result. No, absolutely not. You should take the accurate quantity for the accurate result. Most importantly, you need to follow the direction. You should not find any type of shortcut. This chemical is available, first I will put that, then later on I will put jo dur rakha hua hai. It should not be followed. Then I told you already that you are not allowed to take your food, and if sometimes while performing practical you feel thirsty, you should go outside to the laboratory. First of all, in our laboratory we don't allow you to carry anything else than your rough journal and compass. So you are coming inside with that. If you are feeling thirsty, so you have to go out as you, if your bags are kept outside only. So it is not allowed to eat or drink in the laboratory. A strict action will be taken against the students taking food inside. Now we all know that in our laboratory, in our college, each and every place, we are under the CCTV. Now first aid. Sometime after taking all the necessary precaution, uh, any unwanted scene can occur. So if it is a burn, so immediately we need to flush the cold water until the burning sensation is lessened. Now see, it may happen that it is a severe burn. So we immediately, first of all, anything happens in the laboratory, it is your responsibility to immediately inform your teacher. And if it is a burn, for if teacher is not there but you are there, it never happens in our laboratory. But keep in your mind that you have to immediately keep your burned part under the water. Then injury, you are making the slides or taking the session, so it can causes the cuts or the bruises. So first of all, we make sure that we will not touch any open wound without the gloves, because you know that blood of other human is a biohazard. Then press directly on the minor cut will stop bleeding in the few minutes only. Means if it is a small cut, a small blade cut is there, so it can stop if you will press that particular area with the cotton. Apply cold compress to bruises to reduce the swelling. Then something has splashed in your eyes, so that time immediately flush eyes with the plenty of water for the several minutes. For foreign object lodged in the eyes, do not allow eyes to be rubbed. So these are the certain cares we will be taking inside the laboratory. Now next lecture I am going to explain you about the use of safety symbol. That now this is all about, in general I discuss what are the good laboratory practice and what are you going to follow to maintain the good laboratory practice in our laboratory. So good laboratory practices we have discussed on the industrial level, and as well as on the country level we have discussed what are the authority bodies. The point to be remembered is GLP, full form good laboratory practices. OECD, full form is Organization for Economic Cooperation and Development. NGCMA, full form is National Good Laboratory Practice Compliance Monitoring Authority. And the most important, you need to remember, GLP certification valid for 3 years, and after 3 years you need to go for recertification, and it is awarded for the physical uh, chemical testing, toxicity study, mutagenicity study, environmental toxicity, residual studies, studies on effect of the natural ecosystem, analytical and clinical chemical testing. Then after, what you have to follow, I hope you will never ever forget in the laboratory. So for today, at this note, we'll stop. If you have any doubt, definitely you can ask me.